In this lecture, we're going to talk about exactly how cells harvest energy in order to do their work. So this is what we call cellular respiration. Before we talk about how organisms are going to harvest energy on the cellular level, we're going to take a look at the broader kind of picture of producers versus consumers. So we see here that light powers life. Sunlight is what gives energy and life to this world. And so light energy from the sun is going to power these chemical processes that make all the organic materials and molecules that we have here on Earth. And we're going to get to that in the next lecture because that process is what we call photosynthesis. And hopefully you recall from general biology that plants and other photosynthetic organisms are the ones that carry out this process of photosynthesis. When we talk about living organisms on this earth, there are basically two kinds: producers and consumers, or what we call autotrophs and heterotrophs. Autotrophs are the plants and other self-feeders, we call them, because they can make their own organic matter from the power of the sun. And so those are what we call producers or autotrophs. Now, heterotrophs are what we call, or consumers, are organisms that cannot make their own organic molecules from the sun. They have to get their organic and inorganic substances from, eating other organisms. So that's what we call consumer or a heterotroph. So in the picture on the right, we've got a porcupine that is a consumer because he has to get his energy from the berry and plants around, which are autotrophs or producers. So in nature, it's this big cycle of organic and inorganic molecules from autotrophs that are using them in order to make glucose and food for the heterotrophs that are breaking down those molecules for energy and cycling them back to the autotrophs. So the autotrophs that are, or the producers that are performing photosynthesis are using carbon dioxide and water, in order to make glucose and oxygen, in which those heterotrophs are going to utilize in order to have energy for themselves, and they're going to break that down into carbon dioxide and water. So again, it's like this big cycle. Life is just the big cycle of carbon dioxide and water going from one organism to another. Okay, so the substrates of cellular respiration become the products of photosynthesis and all that stuff. So and throughout this whole process, energy is being made and energy is being used by organisms in order to sustain life. So let's start taking a look at cellular respiration, exactly how cells are going to break down the food that they eat to get the energy that they need in order to survive. So for any of this to happen, the cells are going to need oxygen, in order to break down the fuel, the food that we eat. So this is what we call an aerobic process. Okay, so if you do an aerobic exercise, you're breathing in lots of oxygen, and that oxygen is going to be used by your cells in order to break down the food you eat so that you have the energy to do the exercise. So again, it's all a huge cycle. Now, the cellular respiration is going to require an exchange of two different gases. The oxygen that we talked about that we have to breathe in, and the carbon dioxide that we're going to see is a waste product of cellular respiration. So the cells are going to, at the cellular level, exchange those two gases. And that's why, as a whole organism, we need to breathe out carbon dioxide and we need to take in oxygen. Carbon dioxide is the waste product of all of our cells breaking down food and using it for energy. And we need that oxygen as we breathe in order for the cells to do that process. So here's the overall equation for cellular respiration. We've got glucose. That's the main fuel for cellular respiration. That is the fuel that your cells are, best suited to break down to get the most energy from. We'll see later that we can use the other, parts of the other macromolecules that we eat, like amino acids and lipids, for fuel, but it's not the ideal. It's not what your body is actually have been created to do. Now, the cellular respiration is, like we said, going to require oxygen. And so we have our reactants for cellular respiration is the glucose we eat and the oxygen we breathe. So those are our reactants. And as those two things are, kind of broken down, we get our products, which are carbon dioxide, water, and energy. Okay, in the form of what we call ATP, or adenosine triphosphate. So here let's take a big picture look at the role of oxygen in cellular respiration. Oxygen is a key because during cellular respiration, we're going to transfer both electrons and hydrogen atoms from the glucose that we eat to the oxygen so that we can make the water and the carbon dioxide and energy that we get from cellular respiration. So oxygen is a very key element in the cellular respiration process. When electrons and hydrogen atoms are transferred from glucose to oxygen, it's what we call a redox reaction, or that is short for oxidation reduction reactions. So there's kind of two steps. We have an oxidation, which is the loss of electrons, and we also have the second step, which is a reduction, which is the acceptance of electrons. So in this redox reaction in cellular respiration, the glucose is going to lose the electrons, okay? So he's going to become oxidized, and the oxygen is going to gain the electrons, and so he, therefore, we say is reduced. So we have a redox reaction, or oxidation reduction reaction, where glucose is being oxidized because it's losing the electrons and hydrogen ions, and oxygen is being reduced because it is gaining the electrons and hydrogen ions. When redox reactions occur in the cell, we get the release of energy. So as the hydrogens and electrons are going from glucose to oxygen, lots of energy is released. And so the purpose of cellular respiration is to harness that energy so that cells can do work and the organism can survive. And so cellular respiration is going to unlock this chemical energy in small steps. And so we're going to take a look at each of those steps. So the three small steps in which cells are going to harness this energy from redox reactions are what we call the three stages of cellular respiration. And they occur in different parts of the cell itself. We have glycolysis that occurs in the cytosol, or the cytoplasm. We've got the Krebs cycle, also known as the citric acid cycle, that occurs in the mitochondria. And then we have the ETC, the Electron Transport chain, which if you take a look at the picture on the right, it happens in the mitochondria, and it's where we're going to harvest most of that ATP from the redox reactions. We are going to first take a look at the first step, called glycolysis. Glycolysis is going to occur in the cytoplasm of the cell. And in this step, we are going to take our 6-carbon glucose molecule and we're going to split it into 2 smaller 3-carbon molecules called pyruvic acid. And if you take a look at the picture on the right, it's going to show you how much ATP is both used and gained from this process, because in order to break up a glucose chain, we have to use energy. Okay, so 2 ATP are used in order to break up the glucose chain, but as we change the pyruvate within the glycolysis process, we also get a total of 4 ATP in the end. So we have a net gain of 2 ATP during glycolysis. So we can say that glycolysis is going to produce 2 molecules of ATP. And remember, ATP, or adenosine triphosphate, is the molecule that it's the energy molecule in the cell. It's what the cell uses in order to live, survive, do the chemical reactions it needs in order to have the organism survive. You'll see as we go through cellular respiration and then the next lecture, photosynthesis, we actually have to learn this in pretty good detail, although lucky for you, not as much detail as somebody who's majoring in biology. But it's pretty good detail, so you're going to want to make sure that you take notes and draw pictures and stuff like that. It's going to give you more of an understanding of these processes. So as we look at glycolysis, you can see that, again, remember we used 2 ATP, we got 4 ATP in the end, so we have a net gain of 2 ATP molecules. But you'll also notice that we've got a couple of other molecules that were reduced in this reaction. So let's take a look at that. All right, so the high energy and the high energy electrons broken, from the broken carbon bonds, is used to reduce a molecule called NAD with a positive charge. NAD is nicotinamide adenosine dinucleotide. Okay, so long name, we'll just call it NAD with a positive charge. And so these electrons jump from the carbon chain to this molecule of NAD+, and it reduces it down to NADH, okay, because remember, a hydrogen goes along with the electrons. So we get this, a production of 2 molecules in glycolysis. We get 2 molecules of NADH, and we'll see how those are important later in the cellular respiration process. Okay, so far we've only made a net of 2 ATP, which is really not much. Our cells will need, way more ATP than that in order to thrive. So most of the energy, luckily, is still in the 2 pyruvic acid molecules that were broken up. Okay, so most of our energy is still bound up in these 2 molecules. And so the energy then is going to be harvested in what's called the Krebs cycle or the citric acid cycle, and that occurs in the mitochondria. So let's see how that works. So the pyruvic acid that's made in the cytoplasm has to then be transported to the mitochondria. But before it gets into or gets to be able to participate in the Krebs cycle or the citric acid cycle, we have to kind of dress it down a little bit. We have to turn it into a 2-carbon compound called acetic acid. Okay, so that happens here. So we take off a carbon dioxide from the pyruvic acid, and it becomes what we call an acetic acid. And as it enters the Krebs cycle, it is joined with a molecule called coenzyme A to make what is called acetyl coenzyme A. Okay. So that is kind of how it enters the Krebs cycle. It has to enter the Krebs cycle as a molecule of acetyl coenzyme A. And you'll notice that as coenzyme A joins the acetic acid, again, we get a reduction of NAD to NADH. So we're going to get some energy there, and I'll tell you how that works in a little bit. Okay, so thus far we've turned glucose into 2 molecules of pyruvic acid in the cytoplasm. Those pyruvic acids go into the mitochondria, and in the mitochondria, the pyruvic acid is turned into acetyl coenzyme A. All right, so in this slide is kind of just a reiteration of what I had mentioned in the last slide, but now it's here for you guys to write down. So remember, the pyruvic, the pyruvic acid is changed to acetic acid by removing a carbon atom in the form of carbon dioxide. And as the acetic acid turns into acetyl coenzyme A, we get the reduction of NAD plus to NADH. Okay, so let's take a look and see what happens to this acetyl coenzyme A molecule. All right, so now we can get our acetic acid, or the 2 carbons from our original pyruvic acid, which remember was from the glucose molecule. So now we've got 2 carbons from the original glucose molecule ready and set to get into this Krebs cycle. So the coenzyme A molecule is going to attach itself to the acetic acid. It's going to act like a little guide to guide it into the Krebs cycle. And in the Krebs cycle, the acetic acid is joined to a 4-carbon, what we call acceptor molecule, and that is right here. Okay, so now we've got another 6-carbon molecule, but this is called citric acid, hence the name citric acid cycle. Okay, and as this citric acid is being broken down, we get the output of all of these things right here. So we get the final 2 carbons, if you want to think about the final 2 carbons in our original pyruvic acid molecule are now being broken down into carbon dioxide waste. Okay, and as we do that, we get ATP, we get a couple more molecules of NADH, and we also get what's called FADH2. So we'll talk about that in the next slide. So in this citric acid cycle, or the Krebs cycle, we get just a small amount of ATP formed, okay? But we get much more NADH, okay, molecules, which basically are going to be hydrogen ion transporters into the ETC, which is going to produce, remember, like we saw, a lot more energy. So these guys are kind of carrying that energy to the next step. So they are important. We're not losing any energy, but we are kind of just holding that energy until that last step where we can get a big bang. We've also got a second electron carrier, which is called FAD, and that's reduced during the Krebs cycle to make FADH2. Okay, so we get these 3 energy type molecules out of the Krebs cycle, and the starting molecule, or that first 4-carbon acceptor molecule, is recycled or reformed. So each acetic acid is joined to that 4-carbon chain to make the 6-carbon citric acid, and then 2 of those carbons are taken off to form the carbon dioxide, and then we get the rest of the chain just recycled all the time. Okay, so it'll join, another acetic acid that's been broken down from pyruvate, and this whole thing just circle, cycles over again. Okay, so now at the end of the Krebs cycle, we have all 6 carbons that were originally in the glucose molecule now off as carbon dioxide waste. But remember, as we broke each of those off, we got lots of energy. Okay, we got lots of energy from those broken bonds, and right now that energy is kind of held in the NADH, the FADH2, and a few ATP molecules. So let's take a look and see what happens with those molecules. Alright, so like we said before, in glycolysis, in both glycolysis and in the Krebs cycle, we got that NAD plus reduced down to NADH. So in the electron transport chain, okay, this is kind of big picture here. So in the electron transport chain, those NADHs are now oxidized back to their original NAD plus. They're kind of recycled, and they can go back out into the glycolysis area and the Krebs cycle area in order, to get more electrons and hydrogens. And so it's this big cycle here. So in the electron transport, that NADH is oxidized back, and the electrons that it's carrying, okay, are passed to a chain of proteins, okay, like we see here in the picture. This chain of proteins, which is what we call the electron transport chain. So it's like you can visualize the NADH, coming and just kind of dropping off those electrons within the transport chain, and then going back to get more from glycolysis and Krebs cycles. So again, these high energy electrons are passed from the NADH to the electron transport chain. All right, so it gets just slightly more complicated because you would think the high energy from the electrons being dropped off from NADH would actually be used to synthesize or make ATP, but guess what? They're not. Of course they're not, okay? It's much more complicated than that. The energy from those electrons are actually used to move hydrogen ions across the inner mitochondrial membrane. So hydrogen ions are going to go from this side of the membrane over to this side of the membrane. Okay, and the way that we do that, they're being transported against their gradient, so we have to have energy, and that energy is coming from those high energy electrons within the inner mitochondrial membrane there. So they're kind of like, the electrons are kind of helping the hydrogen ions being sucked from, through the membrane there. So we haven't made any ATP yet, okay? We are just moving hydrogen ions from one side of the membrane to another, okay? So it's quite a complicated process, but let's see and figure out how ATP is actually made, because that's what we need. We want to get ATP, okay? So let's take a look. Okay, so this electron transport chain sets up what's called a gradient of hydrogen ions across that inner mitochondrial membrane. So the hydrogen ions are going from one side to the other. And so now we have a high concentration of hydrogen ions on this side. And so we've got a lot of potential energy here just stored in all of those, that high concentration of hydrogen ions. They're going to want to, go somewhere. Okay, so they're going to have high energy. They're going to be like, oh, I need to get out of here. Okay, and so we're actually going to use that energy in order to make ATP. All right, so we got a bunch of these hydrogen ions, like we said, on this side that are freaking out. They're high energy. They're like, oh, I need to get out of here. There's, they're claustrophobic or something. They, can't stand being on that side. But guess what? They're trapped because they can't just diffuse through the lipid, layer here. So what has to happen? Well, we have to have a protein complex, within that lipid, bilayer of the membrane in order for the hydrogen ions to slip through. Okay, and so this is what we call that ATP synthase protein. Okay, that's this one right here, that basically channels all of that high energy hydrogens back through it to the other side, where they're like, oh, okay, I can do this. This is, there's lots less of us over here, and we can handle this. So, that again, the high energy hydrogens on the inside of that have to go through the ATP synthase protein in order to get back to the lower concentration. Okay, so this is where ATP is harnessed, if you can see here. This is exactly where ATP is harnessed. So as hydrogens are wishing through this ATP synthase protein, okay, it is that energy is used to fuse a phosphate to ADP to make ATP now. So that's what we see written here in this first bullet. Okay, that the electrons move down the concentration gradient through the ATP synthase channel, and that energy is used to make lots of ATP. Now you might ask, okay, what's happening with the electrons as they move down, from through the electron transport chain, okay, which are made up of a bunch of proteins. Well, that's where our lovely oxygen comes into play. Okay, the oxygen has to basically kind of catch all of these electrons as it goes down the electron transport chain. So oxygen is going to be catching a bunch of these electrons and fusing with some of the hydrogens that are on this side in order to make Dun, 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 dun. Water. Okay, so that's why we need oxygen, and that's how we get the water that is produced as a byproduct of cellular respiration. Now, on a little side note, you've probably heard of the poison cyanide. Okay, cyanide works by getting into your cells, into the mitochondria of your cells, and actually blocking the electrons from joining oxygen. Okay, so the oxygen can't be utilized, water can't be formed. If it gets too clogged up, then these hydrogens can't go across. So we have no potential energy over here. We've got nothing going through the ATP synthase. Basically, your cells shut down. And if your cells shut down, you shut down. And if you shut down, you die. So that's why cyanide is so very dangerous. So stay away from cyanide. All right. We made it through that long and complicated process. I would suggest making lots of pictures, drawing this out lots of times, and really getting this kind of cycle, where it happens, how it happens, what happens, into your brain before the midterm. Okay, so we're just going to do kind of a quick summary in the next couple of slides just to make sure that we get it. So again, cellular respiration has 3 stages. First stage is what we call glycolysis. Right, that happens in the cytoplasm of the cell. So here we have glycolysis happening in the cytoplasm, or what we call cytosol, of the cell. Starts off with glucose, and we end with pyruvic acid, right? We have, just 2 ATP being generated, and some NADH, molecules as well. Next, each pyruvic acid molecule gets into the mitochondria, okay, where it's going to get dressed up to go into the Krebs cycle, right, which happens in the mitochondria. And this is where all of the rest of the carbon atoms are turned, into carbon dioxide, okay? And as we break up the pyruvic acid into carbon dioxide, we release lots of energy in the form of electrons, and those electrons are captured by NAD plus to make NADH. So in the Krebs cycle, we get just 2 ATP molecules, but we get lots of electrons trapped in the form of NADH and FADH2. Okay, so that is the Krebs cycle. So far between the glycolysis and the Krebs cycle, per glucose molecule, we've only made 4 ATP. So not very efficient, but we're not done yet. Because now we come to our electron transport chain, okay, which remember is between the inner membranes of the mitochondria little organelle. So in this, we get all of that FADH2 and NADH dropping off their electrons into the transport chain, okay, which remember is just a bunch of proteins, which thankfully you don't have to remember their names. They drop them off with those proteins in the electron transport chain and get reduced back to NAD plus and FAD. And those the high energy in the electron bonds are being kind of removed in small steps, where the at each step the electrons are passed in the electron chain. The hydrogens go from one side of the membrane to the other side of the membrane. All right, so the movement of hydrogens across that inner membrane of the mitochondria is going to build up the hydrogen ion gradient. And remember, those hydrogen ions have to move down their concentration gradients from high concentration to low concentration, just like diffusion. And the way they do that is they have to go through that ATP synthase protein. And as they do that, the energy from going from high concentration to low concentration is harnessed, and we get lots of ATP being made. Okay, in fact, we get about 28 ATP molecules being made. And so in the end, we've got, right here, we've got about 32 ATP molecules being made in total. So that is per glucose molecule. Okay, we've got 32 ATP being made total, and that number, just to be honest with you, that number kind of varies between 32 and 38. So we are going to go with your textbook that says 32 ATP, made per glucose molecule. Although I remember it being 36, and this picture here says 38. So there's kind of a discrepancy there as to what scientists are agreeing upon. But for you guys, test-wise, knowledge-wise, know that it's a crap load of ATP. It's 32, okay? 32 per glucose molecule. So one glucose molecule is broken down, and in the process, we make 32 ATP. All right, this is kind of a cool slide because it shows you how your body can use other macromolecules besides polysaccharides, which break down eventually into glucose. So that's the general way. We've got our food made of polysaccharides. Those are broken down into simple sugars and go through the glycolysis. The glucose goes through glycolysis. Well, what about the fats you eat? Yes, fats can be used for energy. It's not the main source of energy, but if your body is running out of glucose, it can switch to fats, and this is where fats can come into play. Remember, fats are made up of fatty acids and glycerol, so they can be broken down. The glycerol can be changed into a form that can go to glycolysis, okay? Or those fatty acids can be kind of pumped in and used for at the acetyl coenzyme A process of the Krebs cycle. So, that's how fats can be used. If it goes, the fatty acids go through acetyl coenzyme A, then we've lost a few ATP in the process, but we can still get energy because it'll go through the Krebs cycle and electron transport chain. Now the proteins that we eat, okay, can also be broken down to form energy, and this is a lot more complicated process and actually uses more energy to get it there than what we can actually get out of it. So it's not as an efficient process as using sugars and glucose, but again, it'll still work if your body is starving. So we can break down amino acids actually to almost any stage, and it can be utilized in almost any stage. So some of the amino acids can be converted to a molecule that can go through glycolysis. Again, some can join up at the acetyl coenzyme A1, and some just can go directly into the Krebs cycle. So there's a variety or a versatility of cellular respiration, which is a beautiful thing, because that means that if our food supply runs low on glucose for any instant, or if our bodies are we can't eat for a few days and our bodies have run out of the glucose, it can convert the fats that are stored in our bodies to energy, and it can also convert some of the muscle, if it's desperate enough, some of the muscle that we have in our body into energy as well. All right, we are almost done. We just have to talk about one more concept, and that is cellular respiration without oxygen. Okay, so I'm not talking about going to the moon and surviving without oxygen or whatever. I'm just talking about, quick bursts running, doing, heavy lifting exercise, that kind of thing, where you may be breathing a lot, but your body is not getting enough oxygen for your muscle cells to harvest all of the energy from glucose with all that oxygen. So luckily, your cells still can make a little bit of ATP without oxygen. It's just not as efficient, but it can help you survive, and your muscle cells go through small bursts of energy. So The muscle cells will do this by what's called fermentation or anaerobic respiration, okay, which anaerobic means without oxygen. And if we think about the 3 stages of cellular respiration with oxygen, it's basically only the first stage, the glycolysis stage, that occurs in fermentation or anaerobic respiration. So in glycolysis, remember, we get the glucose molecule being broken down to the pyruvic acid molecules, and we can get 2 ATP from that. Okay, so 2 ATP is better than no ATP, although again, it's not as efficient as the 32 ATP that we get if there's oxygen around. Now, pyruvic acid cannot go into the Krebs cycle because there's a lack of oxygen. So what your body does is that it has to basically turn it into lactic acid. And you've heard of lactic acid before, I'm sure, especially if you're an athlete, because that's what makes your muscle cells sore. Okay, because your body can't, your cells directly cannot do anything with the lactic acid. So if you're going through a lot of anaerobic respiration, that lactic acid is going to build up in your muscle cells, and it is just going to stay there and kind of just cause irritation and pain. Fun. Until your cells, transport it out into your blood, it goes to your liver in order to actually break it down, back down into a usable sugar. So that's what happens in fermentation or anaerobic respiration in us humans and some higher order animals. Now the other like microorganisms can also do what's called anaerobic respiration. So let's take a look at them. Okay, so small microorganisms like yeast, okay, are able to produce ATP in the absence of oxygen as well. And instead of calling it lactic acid fermentation like we have, in us humans, we call it alcoholic fermentation, because we're going to see that the end product is an alcohol. So it goes through the same glycolysis process where we've got a glucose molecule being broken down into 2 pyruvic acid molecules and a release of 2 ATP for the cell to use as energy. Now, here's where the difference is. Instead of turning it into lactic acid, it will turn it into ethyl alcohol. Okay, so it releases some carbon dioxide molecules and breaks apart the pyruvic acid into 2 carbon molecules called ethyl alcohol. And in fact, this is where the alcohol content in alcoholic beverages like wine and beer is formed. Okay, it's basically to make wine and beer, you have to go through a fermentation process, which basically means you add microorganisms like yeast to it, and the yeasts are going to break down the sugars in the wheat and the barley that are used in the alcoholic beverages to make ethyl alcohol. Okay, and so that's how we get alcoholic beverages. So that, my friends, is all. We are done. Finally. That is cellular respiration. And yes, you need to know that much detail. So study hard, my friends.